So we were going through the citric acid cycle, and we're taking our food sources, our potential energy, and that was glucose, and we're converting it into other energy sources. And we are to the point at which we're creating the major energy source, which are the electron carriers. Okay? And the electron carriers that we are creating are NADH and FADH2. And so we saw the molecule succinate be converted into fumarate. And what did I say about the star? It's important. Okay, now when you look at fumarate, what do you see about it? Symmetrical. And so this symmetry, once the product diffuses out of the enzyme, when it lands into the next enzyme, it can orient itself two ways. And so if we are numbering or counting where the atoms are, the top two can be the bottom two, and statistically the top two are half of the time. If you name them 1, 2, 3, 4, the top two are, the top one is 1 and 4, 50% of the time 1, 50% of the time 4. The second one would be 50% number 2 and 50% number 3. You couldn't distinguish that. All right, the next reaction is it's called fumarase. Okay, that is a common name, so it doesn't give you an idea about what kind of class that particular enzyme is in, but you can see where the reaction occurs. Now, this is probably one of the more simple reactions in that the R groups are small, okay, and so you can identify what's happening, okay, and so what we're doing is we are adding what across the double bond? Effectively what? Water. Well, you see, that's a reactant, right? We see OH and H. HOH gives you dihydrogen monoxide, which is water. Yes. Okay, so how would I go about starting this particular reaction? 92% of the time, what is it going to start off with? A base. Okay. Now, do you see something that has more hydrogens than in the product? Which of these entities has more hydrogens than in the product? The reactants, and there are two. This right here, oops, sorry. Yes, it's the water. This doesn't have more. This has less, right? But this one does have more. I have 2. Here's the oxygen, only 1. All right, that gives me a pair of electrons, and it's going to attack. All right. Now, for sake of argument, does it attack here or does it attack there? Really doesn't matter because they are both the same, but in the enzyme, one of them will be oriented towards this hydroxide, okay? The hydroxide will attack, and then this will kick up and pick up a proton. At this moment in time, we do have a stereogenic center. It is now distinguishable, okay? And so it is not symmetrical anymore. We get what is called L-malate. All right? Now, what is interesting about the citric acid cycle, all right, is different than glycolysis. If you looked at the two cycles, how would you generalize their differences? One's a circle, and one is linear. Okay? So in citric acid cycle, we have to go back to the beginning. And we are one reaction away from the beginning. All right, if you take a look at the classification of enzymes, this is malate dehydrogenase. A dehydrogenase, if you are and should be writing in that little section about what the mechanism should be, you deprotonate something and a hydride will fall off, you must have an electron acceptor. So when you see that name, that gives you the idea. And in this case, if I take a look at the reactant and the product, we have reaction around this bond where it once was a hydroxide with a hydrogen. Here is a ketone. No hydrogen, no hydrogen. All right. Now, I have two atoms. I have no idea what color that is. What is that color? Pink and red. All right. Which is the one most likely to be deprotonated? Okay, let's do vote. How many like red? How many like pink? Okay, a lot of people like pink, yes. The reason the hydrogen is more acidic is that if I actually take it off, the intermediate would have a negative charge on an oxygen. Okay, that is high energy, but not too bad because of electronegativity. If I deprotonated the other, I would have a carbanion. That's usually not good unless it is alpha to a ketone. All right. Is this red hydrogen alpha to a ketone? No. It is alpha to an acid that has a ketone in it, okay? But the acid is in resonance. And so I can't resonate an additional electron in the resonance, so alpha carbons to acids are not acidic. Alpha carbons to a ketone or an aldehyde are. All right, so I know where I'm going to take my hydrogen. I will take the electrons to form the ketone, and I will accept electrons into the NADH. And I have oxaloacetate, which was the first reaction that reacted with acetyl-CoA. Where did acetyl-CoA come from? From a complex, yes. A what complex? Pyruvate dehydrogenase complex. It came from pyruvate. And where did pyruvate come from? Glycolysis and glucose originally. And so we're back at the beginning. Overall reaction. All right. For each round of acetyl-CoA, of which if I do my stoichiometry from glucose, I have to do this twice. Okay? And if I take a look at it, this is where all of the energy is at the beginning. It's bound up in the bonds of the acetyl-CoA. And at the end, energy is here, energy is in this pool, and energy is in this pool. Now, many times, you will not see this in calculations. What do they make this equivalent to? What does everybody kind of make it an equivalent to? ATP. The reason is, if you have an ADP running around, and you make an GTP, what happens is you get GDP plus ATP, and the energy value of that transition is 0. Okay? And it's just an equilibrium. All right, and so it equates to an ATP. All right? So I have extracted all of the energy from the glucose, and at this moment in time, how many equivalents of ATP have you made? All the way from where you started in glucose? 4. Okay. 5. How many glycolysis? 2. Okay. How many for each acetyl-CoA? 1 equivalent for each, and there are 2 that come from glucose. So that's got 4. The transition in the complex, the pyruvate dehydrogenase complex, did you get any ATP there? No, but you did get an energy source. Do you remember what you made at the end of the pyruvate dehydrogenase complex? I went from a 3-carbon intermediate to a 2-carbon intermediate. One of the things that I made was CO2, but CO2 has no more energy. And I went through the whole complex reaction. I ended up with NADH. Okay? And so there was some energy extracted at that moment in time. All right. Oh. Okay, I'm subtle. Okay. All right, how do we turn this on and turn this off? We turn all of this on and off with lots and lots of basically allosteric effectors. Okay. In this, there are some things that you probably will see a pattern. Okay. Let's take a look at one of the patterns. Okay. ADP, 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 AMP. Okay, what does ADP and AMP represent inside the cell? It represents energy. Does it represent high energy or low energy in the cell? Low energy, okay? So it seems that every time I see a molecule that represents low energy, ATP being used, I speed this process up. They're all positives. Why? Because at the end of this, in the next step, I'm going to use that energy pools to make ATP. Okay, so that seemed reasonable. Okay, let's see something else. Check this out. ATP, ATP, ATP. ATP represents energy in the cell too, but what did ATP, if that is high in concentration, what happened to this process? It slows it down. Okay, it says, don't break down these potential energy pools if I don't need to synthesize ATP. Does that seem reasonable? Okay. All right. Let's try some other things. All right. Succinyl-CoA will slow down this enzyme right here. Okay. Why would that happen? Or describe what kind of inhibition that is. Okay. It is called feedback inhibition. If by chance you make a product and the product goes high in concentration, that means the other steps aren't running. And if the other steps aren't running, I'm building up this concentration too high, I'll say, stop it. Does that make sense? All right.
Is that not the same as the ATP going in concentration?
Yes, but the ATP is not directly in the pathway. It is several steps back. It is a type of feedback inhibition. We usually talk about ATP and ADP as a measure of what we call energy charge, or it's called energy charge. But it is similar in effect. Usually we talk about feedback is one pathway, one of the intermediates in the pathway goes back and feeds back into that pathway itself. All right. Oh, wait. I forgot one. ATP slowed it down. Okay. There. All right. Oh, here was succinyl-CoA. That feeds back and stops that one. Okay. All right. Acetyl-CoA here. If it builds up here, that means it's not entering the citric acid cycle. That feeds back and turns what? That off. It's the enzyme that produces it. It turns it off. All right. If by chance, now this is getting all kind of messy, okay? If by chance, I'm going to back off for just a second so I can see some other stuff. I don't know how many backs I have to do. All right, some other ones that feed back. All right. Here, NADH and NADH and NADH and NADH all do what? Turn it off, because the product of what this pathway does is to make NADH. So if I have a lot of it, I slow down. The opposite is this. Use the opposite color, NAD+. If I have a lot of NAD+ roaming around, I have the capacity to grab hydrides, and so that means speed it up. All right. CoA. CoA is a reactant to convert pyruvate to acetyl-CoA. If I have a lot of CoA, the starting material, and a lot of pyruvate, the starting material, it makes it go fast. Okay. What on this side, right here, doesn't quite make sense to what we have talked about? Yes? Yep. Technically, in this case, they would be feedback because they are a part of this particular pathway. Yes. What is one thing on this chart that is speeding things up that we haven't talked about at all? Okay. Why in the world would calcium speed up the production of ATP? Okay. Is there any tissue type that you know of in your study of biology that is excited by calcium? Muscle. Muscle and heart, both of them. And for muscle to go, what does it need? A lot of ATP. And so in this case, this is a regulatory event for the citric acid cycle in muscle. And so when calcium is released, which starts the myosin and actin contraction, which uses ATP, it's also a great signal. Hey, I'm about ready to use a lot of ATP. Let's start running the citric acid cycle to make ATP. All right. There's a whole other pathway here that I haven't shown you yet. They're going to talk about regulation. Okay. I didn't show you the reaction that makes pyruvate go into oxaloacetate. Okay. In this case, it's important for us to be able to do this. This is the reason. Remember, the citric acid cycle is both degradative and synthetic. So what does that mean? That means a lot of these molecules get taken out of the circle. If you take a lot of these out of the circle, you don't have enough to feed back here. If you don't have oxaloacetate, what happens to the whole circle? It stops. So we have to have an alternative way to get oxaloacetate, and we can actually take pyruvate and make oxaloacetate and pyruvate to make acetyl-CoA, and we can actually start the pathway just with pyruvate itself. All right? And so we are going to look at that particular process. Another reaction that I wanted you to be aware of, we've pointed this out before, but the alpha-ketoglutarate dehydrogenase complex is simply a different specificity, but the same mechanism as the pyruvate dehydrogenase complex, which uses the same cofactors. You need to know the cofactors that are used in pyruvate dehydrogenase complex. What are the cofactors that are used? There's lots of them, and they're all acronyms, so it's easy to remember. FAD was used, yes? NAD and NAD plus and NADH is used, yes? TPP. Lipamide, that is correct. And There's one more. What does the carbons get attached to from the pyruvate to go into CoA? 5 major cofactors to do that reaction. Okay, there is a disease that is seen in the production of, or the reactions with, alpha-ketoglutarate and pyruvate. And it lacks vitamin B1. Okay, so if you lack vitamin B1, you cannot make TPP. Okay, and if you do not have TPP, you cannot convert pyruvate or alpha-ketoglutarate into the next step. So what happens is the concentration of both pyruvate and alpha-ketoglutarate go very, very high in the blood system. Okay? You also cannot extract energy as well. This kind of disease is seen in the Far East because a lot of their caloric intake is rice, but they tend to eat polished rice. The vitamin B1 is found in the husk. And so when they polish or take the husk off, they have plenty of calories, but they don't get this particular vitamin. But here in America, we're all good because if you have cereal, you look on the side and you see all sorts of vitamins, right? We actually, in our processed foods, at least we put some vitamins, okay? And so we don't seem to have that, we don't run into this disease as often, okay? Here is something that people get very confused about. What is the difference between a disease and a symptom? Okay. The symptoms here are high concentrations of 2 molecules that shouldn't be high, pyruvate and alpha-ketoglutarate. Okay. The disease is the lack of a cofactor, which is because you have a lack of a vitamin. Treatment to this is giving the vitamin. Okay, but a lot of people think that get confused about what the disease is and symptoms. In a diabetic's case, the symptom is very high concentrations of glucose in the bloodstream. Okay, that's not the disease. The disease is either I can't produce insulin or I can't process insulin, okay, because there are 2 different types. It's the either not making it or not being able to use the insulin that is there. All right. Remember I gave you stars and there were molecules that were symmetrical? Okay. If you actually look at citrate, not drawn the way it is drawn, but if you look at citrate, at the top there's a methylene and an acid. At the bottom there's a methylene and an acid. So that makes citrate what? Symmetrical. All right. Now, here in citrate, all right, it is symmetrical, and so it gets into the enzyme, aconitase, and then you get isocitrate. And if I labeled these atoms A or B, okay, which atom is at the bottom, A or B? Okay, well, because it was symmetrical, we believed that you probably should see 50-50% in that. But we didn't just think about it, we actually tested it. And how we tested it was we gave animals radioactive molecules to start the process. This is way back when, before PETA got really strong, okay? And so what we did was we allowed radioactive material to come in, and you can see this radioactive material in the citrate. If it was in this particular position, after it was processed, okay, you could have either the C14 at the A position at the top, or it could be flipped upside down, where the A position's here and the hydroxide is on the A. All right? Now, those are the possibilities. What we find is that in every single case, the C14 is far away from this hydroxide. This particular C14 is represented by the acetyl-CoA that came in. The hydroxide is always on the oxaloacetate that comes in. Now, for a reaction to actually have asymmetrical outcomes, all right, you have to have something asymmetrical to do it. And what is asymmetrical in this, the molecule is not asymmetric, but the enzyme is. And the enzyme binds the citrate in only one way, in which it places the base that takes the initial deprotonation next to the carbon, or the carbon that is part of oxaloacetate. And that's how it orients itself inside the enzyme. So we get stereochemistry from this particular reaction by the enzyme itself. Okay? And so we only see this case where this is the acetyl. Oh, well, there's an E in there somewhere. And this is from the oxaloacetate. Okay. There you go. Being subtle. Now, one thing about the NAD plus dehydrogenase, I did show you this reaction in which the hydride comes in, okay? And in this case, we did a little kind of experiment, and we are going to do a redox reaction. The base will take this proton because that's more acidic. The electrons will fall in, and in this case, what is D? Deuterium. Actually comes in, and I get a D that is accepted in the NAD plus to become, at this moment in time, we call that NADD instead of NADH. Now, what is interesting is that you can isolate the NADD and transfer it to another reaction in which the reactant has a hydrogen there. Okay? And then we react it with the NADD that we just produced, and in every case, this incoming reactant, where if I reverse the flow of electrons and I allow that to form, this to fall off, in every case, the deuterium that was added is the exact one that comes off. And so whatever hydride comes into the NAD plus is the hydride that comes off. And that was just an interesting way in which that enzyme works. All right, the citric acid cycle will degrade, but also will produce. And a lot of these smaller molecules will be the substrates to start synthesis of lots of things like fat and amino acids and porphyrin and hemes and pyrimidines. And is the purines over there too? Purines, let me think. No, purines can be made from other sources. But we can start taking lots of things off of there. And so if we start doing synthesis, we could deplete the entire citric acid cycle. That's why we are going to look at this reaction. This reaction allows pyruvate plus a CO2 plus ATP plus water to create oxaloacetate, PI, 2 protons, and 2 ADPs. All right, here's what I need to teach you about this at this moment in time. I say in the reaction I have this molecule. You know what that is? Name it. Carbon dioxide. Okay, great. Look at the reaction. Show me where carbon dioxide is in this reaction.
Between step 2 and 3.
Between step 2 and 3? Okay, oh yeah. There's it for a second. All right. Here is what every biochemist knows, and they never tell you, okay, formally. CO2 is dissolved in water. How much CO2 can you dissolve in water? What's the solubility of the gas? A lot or little? How much gas can you dissolve in it? Actually, not a lot. So what happens is CO2 actually reacts with water to make a theoretical H2CO3. Okay? This molecule has never been isolated. But these two things have. A proton plus what? What is it? I heard something. Do you know what that ion is? Do I need to give you a TRQ from GChem 1? I think I do. Bicarbonate. We don't call that bicarbonate in GChem 2, GChem 1 anymore. What do we actually call it? Hydrogen carbonate. Okay, that's the newer name, but bicarbonate is what we think about, and that's common name. So anytime you see bicarbonate in the system, that is a CO2 that's been reacted with water, okay? And it is also the major component of the buffering system of the blood, because we're always producing CO2 as we break down all of our food product, and we have CO2 running around as bicarbonate all the time. And so in this process, what actually occurs is I can take the bicarbonate, we activate it, we react it with a vitamin that swings it from one active site to the other, and then we react it with pyruvate to make oxaloacetate. This can refund the citric acid cycle, okay, and allow the citric acid cycle to continue to move. This is also a preview to the first step of the synthesis of glucose, which is called what? Gluconeogenesis. Okay, and so this pathway is a double thing. Okay, it actually is a marriage of two different pathways in the body. All right. The citric acid cycle produced what? Lots and lots of, what happened to the carbon? It produced what? The carbon of the pyruvate that went to the acetyl-CoA becomes, after it goes through all the citric acid cycle, we end up with carbon dioxide. Okay? Gives off carbon dioxide. There is a cycle that is similar in plants. Okay? For us, CO2 is a waste product, right? In plants, how in the world could I have a citric acid cycle like pathway? Why is that odd for plants? They what? They use CO2. Do they produce CO2? What's plants' waste product? Oxygen. Okay? So how in the world can plants do or even have a cycle that is similar to the citric acid cycle? And why in the world do we even have a cycle like that? Okay? This particular cycle is used primarily in the germinating portion of when a seed is buried in the ground and begins to grow. Plants get their energy from what, primarily? The sun. And it makes all of their energy. But when a plant in the seed is buried underground, what can't it see? The sun. So, seeds are composed of a large portion of fat. Okay? That fat is intended to provide energy until the plant can get above the ground to start receiving the light. And so this cycle is used to get it through that particular stage. If you look at this, acetyl-CoA can be derived from fat also. You have oxaloacetate to make citrate. That's a citric acid cycle reaction, same enzyme. Citrate becomes isocitrate. That's a citric acid cycle intermediate, right on. And isocitrate becomes, oh, whoa, that's a different reaction. Succinate comes off, which was a citric acid cycle intermediate. Okay, but this is now a waste product from this process. I take in another acetyl-CoA and I create malate. Well, that reaction is not in the citric acid cycle, but malate is a citric acid cycle intermediate. And then malate gets converted into oxaloacetate. That is a citric acid cycle enzyme. And I make energy in the form of NADH, which then can be formed it can be formed into ATP. And so I can use a cycle that has 3 enzymes that are exactly the same, 2 enzymes that are different, with 1 intermediate that is different, a different waste product, and provide energy from fat within plants. Okay? And so, also, this succinate is actually a precursor to create sugar. Because when for it to grow, you have to grow cellulose to grow the stalk. And so the sugar is derived from the succinate rather than the normal process that we do from CO2 being absorbed and fixed to create sugars. Okay? And so this is a wonderful compare-contrast type of question. What looks like citric acid cycle? What doesn't look like citric acid cycle? So I would know a compare-contrast for this. All right. We are done with the lecture material. We are moving now on to the active learning portion of the day. Okay. All right. All right, this type of activity is assigned quite often because way back when, we didn't actually know these pathways. Okay. So in our attempt to identify pathways and how things were converted, a lot of our understanding began with this. They took radioactive sugars and they allowed the process to occur in living systems. And so if you put radioactivity in one atom in the starting material, you would assume that it's going to be in one atom as it gets broken down, and then you can isolate every single molecule that has radioactivity and follow where that radioactivity ends up, and you can start making connections. Oh, this got converted into this, and this got converted into that, and that got converted into this. And it was supposed to be very, very simple, straightforward, and tell us everything. What do you think happened? It wasn't simple. It wasn't straightforward. Okay? And so, what I want you to do is this. I believe the question says this. If by chance you ran a glucose with radioactive carbon-14 and carbon number 1 of glucose, all right, where would it end up in the oxaloacetate at the end of round 1? Okay. Does it say at the end of, let's see, so glucose placed in through the cycle, and what was the fate of the radioactive carbon after one round, yes, after one round of the citric acid cycle. So that is what I'd like you to work on. Okay, you want a bonus? Okay, here's the bonus question to this. If at the beginning of the citric acid cycle, okay, where you left the radioactivity, and another acetyl-CoA adds with no radioactivity at all, where would the radioactivity be in the second round of the citric acid cycle? Okay, so here's what's happened. Think about this. It's in glucose, right? Goes all the way through. It's going to be somewhere in the oxaloacetate. That's the quiz. So basically, you're going to tell me, you're going to draw the structure of oxaloacetate, and you're going to put carbon-14 where it should go. Okay? The bonus is this. If this oxaloacetate that you said where the radioactivity is goes one more time around, but we don't add any more radioactivity, okay, where does the radioactivity end up after the second round? In the oxaloacetate. All right, you have two choices. You could turn in a page that has one structure of oxaloacetate that say end of round 1 and one structure of oxaloacetate end of round 2, and that would be very easy for me to grade because it's either all right or all wrong. Okay? If you don't want to do that, then it would be good maybe to show exactly where it is in each of the steps, and I can give you partial credit to know where you might have gone wrong and at what step. Okay?